I will invite the next speaker to the podium, who is Thomas Moore. He's actually substituting Professor Alke Fink, who couldn't actually come today in person. And it will present about materials science in nanomedicine and materials perspective from the University of Fribourg. It's all yours.
Thank you for the introduction. Anybody who knows Professor Fink, I'm obviously not her, but I'm a postdoc of hers who will be stepping in. And the talk today will be on nanomedicine, but using a materials perspective. So what we do in our group is mainly look at materials characterization and very fundamental questions about particle-cell interactions. So I won't go through this slide. We all know the story, how nanomedicine has been developed over the past 60 years or 50 years or so. But what I want to talk about, there's an interesting paper that came out in 2013 where Vendito and Soka looked at the number of publications for particles coming out, the number of times these papers are cited versus how many clinical trials. And also we've heard a lot of talks about this Warren Chan perspective where they're discussing the kind of failings of nanomedicines at the moment. While there are very exciting things going on, and we're all very excited about this, there does seem to be a problem translating across from the things that we're doing in the laboratory and what's coming out into actual products. And so, as I said, we study very fundamental things. So a lot of the talks here have been about preclinical in vivo data and clinical trials data, but we're focused more here at the in vitro stage where we're looking at particle-cell interactions or particle interactions with proteins and things like that. And one of the things that we kind of hypothesize might be an issue is that particles behave very different from small molecules. So if we look at the standard transfer from preclinical in vitro studies to the clinical trials, you know, with small molecules, you can treat them in a very standard way, but with particles, they behave very differently. So when you put a particle into a complex biological solution, you can see that you immediately have protein absorption generally. If your particles are not coded correctly or if they have some kind of issue, you can have particle aggregation. Certain types of particles can dissolve, which may or may not be a desirable function for you. And you can have detachment of stabilizing ligands on the surface. So we think an issue might be that there's a lack of fundamental understanding when we look at particles and at the nanoparticle cell level, and then also interlaboratory differences between the way we do experiments. And so we can take, as an example, there's a review published by Harold Krug where he discussed the different nanoparticle characterizations. There's also some papers by Peer and Howard where they looked at discussing different characterization techniques for nanoparticles, and especially when we look at a nanoparticle toxicology perspective. And so we are focused here at the nanoparticle characterization in biological media and then comparing between laboratories and published research. And actually, my advisor asked me when I first came to the lab if there were differences in the way we pipette, which I thought was a very silly question because I said, yeah, I mean, I always add, you know, premixing particles, and I didn't think this would have any kind of effect on what the experimental outcomes were. But she asked me to look into this, and so we started to do some experiments. And so we wanted to see, does the way that we actually do particle studies in vitro have an effect on the in vitro outcome, and does pipetting have an effect on nanoparticle colloidal stability? So to do this, we used 90 nanometer gold particles and 15 nanometer gold particles. We coated them with polyvinyl pyrrolidone, and then we looked at 3 different administration methods: concentrated, mixed, or pre-mixed. And as I said, my training, I always did in this kind of way when I was looking at in vitro studies, but if you're doing something like live cell imaging, you may not have the capability to pre-incubate, and you may need to add a concentrated bolus. And so we looked at these 3 different administration methods using J774A1 mouse monocyte macrophages with gold particles. We looked at 3 different coatings, but I'm only going to talk about PVP for this talk, and then did some standard characterization. So we looked at them through TEM. We used UV-Vis to characterize their stability in media, and we also looked at using dynamic light scattering, but also depolarized dynamic light scattering. So if we look at the 90 nanometer particles, this is if we add them to complete media supplemented with 10% fetal bovine serum, if we look at the concentrated administration and premixed administration, you don't seem to see much difference. You don't see a redshift in the peak, and this indicates that the particles are most likely stable. We see similar results with the 15 nanometer particles, but what we do see in data not shown is that the scattering intensity increases over time, and this is indicative of some kind of protein absorption pattern. And then when we use depolarized dynamic light scattering, which is a way to put a polarizing filter in front of your detector in dynamic light scattering. And what this enables you to do is filter out any scattering from organic molecules, and you mainly get the scattering from very intensely scattering species, which would be for us the gold nanoparticles. And so what we saw is that for all of the cases, we did not observe any kind of aggregation, and this gave us the clue that our particles are stable in complete media. And so what this tells us is we can move forward with our studies, and we think this is a very important step in the whole particle development process, is to ensure your particles are stable, because if they aggregate when they go into biological media, then the effects that you're measuring could be from some weird species where you have aggregated particles. So what we did was then, using ICP, we looked at the amount of particles that were depositing and either absorbing onto cells or being taken up by cells. And in this study, I don't make a distinguish in between those two phenomena. But what we saw, which was very interesting, is that the 90 nanometer particles, when we administered them either with a concentrated or the mixed way, so that's when you add a concentrated dose and then maybe mix it up and down, or just add it directly to your cells in vitro, there's a significant difference between how much was deposited onto the cells versus if you just add with a premixed case. And so you can see here, this is the fraction of initial administered particles that we're depositing. And so we saw that this makes a quite big difference. And so then we asked the question, okay, is this a biological phenomenon, or is this just some kind of effect? And so we, from the particles, so what we did was we took the cells and then we incubated them at 4 degrees C to try and minimize any kind of active cellular processes. And what we saw was that this trend still remained, where you have an increase in the concentrated administration absorbing onto the either cell surface or the well plate bottom compared to the premix conditions. When we did this in just water, we did not see any absorption, so we thought that at least with the water case, they're not sticking to the well plates. And then we looked at different concentrations, so at 20 micrograms per milliliter of nanoparticle and 40, and then the other studies were done at 70. And what we see at the different concentrations is, while they end up more or less in the same area in terms of the amount that they're absorbing, we see that this trend continues, where in the initial time points, the concentrated and mixed situation still absorbs more. And so then there's this big story in the nanoparticle field right now, at least BioNanomaterial interactions, is this protein corona story. So what we wanted to look at, and this was just done very quickly, was to see whether the administration method changes how proteins absorb onto the particle surface. And so we saw here that if you had concentrated, you have more protein, about 2-fold increase in protein absorption compared to the premixed case. And so this is just a picture showing how these situations look very different when you add in the different ways. And so for the 15, we did not really observe too much difference, but what we saw is quite interesting, is you see almost a reversal where the premixed is higher, and we couldn't really explain this. And so we asked our colleague in the chemistry department, and there's a whole section in the forthcoming manuscript where we did using the in vitro sedimentation dosimetry or diffusion and dosimetry model. We looked at how the particles are sedimenting or diffusing in solution, and we thought maybe that this could explain what's happening. And so what he did was he modified this ISDD model, and to try and figure out what might be happening, added a component, an expression for nanoparticle aggregation. And what we saw is that this more fitted. So while we were not able to detect this with the UV-Vis or with the DDLS, we did see that perhaps there's some kind of clustering happening where this would maybe explain why we see this phenomena. And so what we saw in the study is that the administration method can have an effect on particle-cell interactions, and it's something that might need to be taken into consideration when we're moving forward with our in vitro studies before we move to the in vivo, and that this is not a cellular mechanism, but it seems to be more about the particles being introduced into a complex biological media. And so then we need to use robust characterization to figure out what our particle identity actually is when it goes into this complex biological media. So I'm a little bit over time, sorry, and I'd just like to thank my collaborators, both at the Adolphe Merkle Institute and also Department of Chemistry. Thank you.
Thank you, Thomas. Thank you very much. Burning question for Thomas. Very basic research. Do I see an end? Okay, yes, please.
Interesting phenomenon. I'm not sure that I'm happy with it, but and did you look at the effect of the media, so buffer ionic strength and those kind of physical chemical characteristics? If so, did you find any effect?
So media is not something that we looked at. We were basically using the media for this cell. And it is curious, maybe if we change the ionic strength of the solution, it's going to have a different effect. And of course, we have seen before, if we change media, particles that were stable in media 1 are going to aggregate in media 2. What was maybe more interesting was that this effect was very drastic with a PVP polymer coating, but it seems to be very, it's minimal if you use, for example, polyethylene glycol or PVA. But the reason we were more excited to use the PVP is because we had more particle uptake. So with a PEG, you have this shielding effect, and then we see very little uptake anyway. I hope this answered the question.
Thank you. Thank you very much. Unfortunately, we had to move on to the